I'm Bob Waterston. I'm a professor of genome sciences here at the University of Washington. I'm also the Gates Chair of Biomedical Sciences. We have a big paper coming out in Science. That's the culmination of several years of work by groups, many groups across the country and even across the world, all on the tiny little nematode Caenorhabditis elegans. Its name is longer than the worm. The worm is only a millimeter long, barely bigger than a piece of dust, and yet it's an animal. It has muscles, guts, neurons, just like people, and it's been used intensively over the last 50 years for study of biology. And indeed, in the last 8 years, three of the last 8 Nobel Prizes have been awarded for work in C. elegans. And that's because even though C. elegans is a little worm and very simple organism, that makes it very useful, very powerful to study in the lab. And so we can make discoveries in the worm, and yet because it's related to people through evolution, it's a deep relationship, but it's got all the same functions that we do. So when we make discoveries in C. elegans, they apply to humans and across all of biology. And so that's why the worm has been studied so intensively. And indeed, because of its importance to biology, it was one of a few organisms selected as part of the Human Genome Project to have its genome sequenced. And I got involved in the Human Genome Project through our efforts on C. elegans. We completed the sequence of C. elegans in 1998, and the methods we devised for that, were indeed applicable to human, and we used that in human along with our colleagues in England and other labs across the country and the world to complete the human sequence and then the mouse sequence and so forth. We had the genome sequence, but the genome is just a string of DNA. And DNA is just made up of 4 letters over and over again. A, G, G, just 4 letters, like letters of the alphabet. But there are 100 million of them that make up the C. elegans genome. And we could partially read what was in that DNA, but only incompletely. You know, the DNA is the instruction set, that information that is used to make a worm. Its genes and all this regulatory sequences and so forth. But we couldn't read that. And so in 2007, the National Human Genome Research Institute, part of the National Institutes of Health, funded these 10 groups to collect experimental evidence that would reveal what the instructions in the DNA really were. Genes are the parts of the DNA that are used to make RNA, and then the RNA is used to make protein, and the proteins are the parts that are the real doers of the organism. So we went after see if we could find all the genes, and we did this through some novel technologies, and the result is that today we have a good understanding of the 21,000 genes in C. elegans. About half of these are different from what was predicted. 1,000 of them are new and weren't predicted at all. And they also produce a wider, more complex variety of RNA molecules than people thought.